All right, folks, we'll jump right in. I'd like to welcome everyone to today's CHIP Landmark Idea Series talk, Prospects for Hyper-Personalized Medicine by Timothy Yu, who's in the departments of the program in genetics and genomics here at Boston Children's Hospital. He's also at Harvard Medical School and the Broad Institute. He is an expert in molecular characterization of patients and the application of genomic sequencing technologies in medical practice, as well as in advancing the genomic sequencing technologies themselves. He's had some extraordinary achievements in hyper-personalized medicine, which we're going to hear about today. The couple of quick logistical reminders. You can put your questions for Tim into the Zoom chat or the Q&A section as we go within the Zoom platform, and we'll collect those for the end. That lets us hit the ground running when we get to the question section. And so, I'm Ken Mandel. I'm going to be moderating today. I direct the Computational Health Informatics Program, or CHIP, here at Boston Children's Hospital. We host the Landmark Idea Series. We're a nearly 27-year-old program in computational health, biomedical informatics, and we have about 30 faculty studying medical informatics from across the spectrum, from the molecular all the way up to care system redesign. This particular series, which you're welcome to join in the future, hosts a wide variety of speakers who have big ideas. We've heard from Nicholas Christakis, Ricky Bloomfield, health lead at Apple, David Clark, an inventor of the internet. One that was off the beaten path for us was Shep Doleman, who took the famous picture of the black hole, who gave a phenomenal talk on team science that we found actually extremely relevant to what we do. You can learn more about these on the chip.org website. So if you want to tweet about this, feel free. You can use a hashtag. We're tagged at @BossChip. Tim, I'm not sure if you're a Twitter guy. Do you have a Twitter handle you'd like to tell us about?
I am on Twitter. I'm on @TimU.
@TimU.
Thanks, Ken.
Thank you, @TimU. And again, we're a program that, by the way, I'm going to give in one quick plug. We have a faculty search out right now, closes for applications on February 1st. And our information about that is available here on the website. So, Tim, I'm going to stop sharing my screen and turn it over to your talk. Again, folks can put questions in the chat, and then there will be ample time for an interactive session at the end.
Thank you so much, Ken. I really appreciate the invitation and this chance to have this conversation with all of you. And I'm flattered to be following in a series of what sound like a series of really wonderful talks. I'm going to have to take a look at this myself afterwards. As Ken said, I'm a neurologist and a geneticist at Boston Children's Hospital. And the title of my talk today is Prospects for Hyper-Personalized Medicine. I'll go ahead and get my screen share up and running for you all. How's that?
Great.
Good. Okay. And it's, as I said, my pleasure to be here to talk about a topic which is really near and dear to my heart. I've been working at Boston Children's now for over 10 years, and having my own independent laboratory here for the last 7 years. This is a project which began relatively recently in the time span of our laboratory, but it's one that has consumed a disproportionate amount of our time, and one that we're really terribly excited about. First, by way of introduction, well, first I have disclosures to share with you on this slide here. But just a little bit about our laboratory and what we're about. Our research group, which is shown, a partial representation is shown here, has been working in the space of neurogenetics for quite some time. Topics that we have focused on are listed on the slide: human brain development, trying to understand the genetic underpinnings by which the brain is built both in normal development as well as in developmental disease. Autism genetics, following on that theme of neurodevelopmental disease, has been a particular focus within that category. And as Ken alluded to, we've invested a significant amount of time developing principles and pipelines and tools for genome analysis and interpretation in the service of pursuing those two broad aims. The talk that I'm going to present today focuses really on a fourth category in the bottom right, on genomic medicine. And this work focuses on some problems that have become apparent to us in this space as we've been working on it for over 10 years, and some new opportunities that represent a celebration of new tools that are available to us as translational researchers in genomics. Now, to set the stage for this, here are a couple of facts. In genetics, especially in pediatric neurogenetics, many of the children that we work with have what is ultimately described as a rare disease. The rare diseases in the US are described as conditions affecting 200,000 people or less. And 200,000 may not seem like a particularly small number, but the reality is that most patients with rare disease have populations sizes, most rare diseases have population sizes much, much less than 200,000, perhaps 1,000 or a few thousand, perhaps a few hundred in many cases, or for many recently described diseases, perhaps only a few dozen. So while individually rare, rare diseases in aggregate affect a large number of people. 350 million patients worldwide are estimated to have a rare disease in statistics compiled by the Global Genes Consortium. And of these, as I mentioned, 80% of these are genetic. These conditions also, unfortunately, disproportionately affect children. 50% of these conditions involve children. And very tragically, 30% of these affected children will die before the age of 5. These are very, very serious conditions. And perhaps reflecting that only a tiny fraction of these, 5% of these, have an FDA-approved treatment. And in many cases, those FDA-approved treatments, while perhaps being approved, aren't necessarily disease course altering, that they may impact the quality or they may affect symptoms, but maybe often do not affect the ultimate trajectory for the condition. So it's against that backdrop that we've been thinking about this problem increasingly, because as I mentioned, the first 2/3 of our laboratory's research really focused on the diagnosis and discovery side of the rare disease puzzle. And by developing tools and applying them to study the genomics of these conditions, we've greatly accelerated our ability to diagnose patients with rare diseases. But that has only, those successes have only highlighted the chasm that still is in front of us, which is that developing treatments for these conditions still takes a really long time. Now, the whole theme of my talk here is focused around a single idea, which is that this gap, this time that it takes to develop a treatment, taking an unconscionably long time, this fact is beginning to change. And no case perhaps highlighted this more dramatically for us than this case, which we had the good fortune to work on over the last 3 years, which we published in the New England Journal last year, which has received some attention, so maybe some of you have heard of it. And in this article, we published an article entitled Patient Customized Oligonucleotide Therapy for a Rare Genetic Disease. We described the ability to develop a drug for an individual patient in under a year's time. And while you may be familiar with this because it has gotten press coverage before, I'd like to take you through some of the backdrop of this as scientific foundations for the concepts that I'm going to be discussing. First, how did this all start? Well, actually, interestingly, back in January 2017, we happened across a post on social media in which my wife forwarded me this message from the Physician Moms Group of Massachusetts. This was a plea from another children's colleague on behalf of a college friend of hers, describing a family who had just been diagnosed with Batten disease, who needed help with a molecular diagnosis. Following that thread, we learned about a young girl named Mila, her name and photographs are shared here with the family's permission, who was in Colorado at the time, and she had suffered several years of insidious vision loss and neurologic decline. And after an extended diagnostic odyssey, which many of us are familiar with, she received a very unfortunate clinical diagnosis from her clinical team in Colorado. She had been exhibiting symptoms of vision loss, ataxia, and cognitive regression. And a skin punch biopsy had shown abnormal fingerprint-like inclusions on electron microscopy, showing lysosomal buildup of something called ceroid lipofuscin. So what are we looking at? These fingerprint-like smudges here represent accumulations of protein and lipid inside of cells that should not be there. And this characteristic appearance indicated that this was a lysosomal component. There's some part of the recycling machinery of the cell, representing the lysosome's inability to clear normal cellular waste material. And this finding is pathognomonic for a condition called NCL, or Batten disease. NCL stands for neuronal ceroid lipofuscinosis, but it's easier to call it Batten disease. And there are 14 different forms of Batten disease, each of which is caused by recessive mutations in genes that seem to have something to do with the function of the lysosome, either in trafficking, formation, biogenesis, or function. And unfortunately, in the absence of proper working copies of these genes, cells with these recycling defects undergo progressive death. And in particular, it seems to affect the central nervous system selectively, such that neurons in the brain and also in the retina progressively die. This gradual cell death occurs over years, and it results in the symptoms described in our patient. And unfortunately, this is such a rare disease, her particular subtype is called CLN7, that there was very little known about this disease, that there was very little biological understanding of this condition. And at the time that we were introduced to her, no one at that point was working on any sort of therapeutic approach at all. And this is a progressive condition that leads to death by around age 11. Now, the wrinkle in this case was that she had only received half of a diagnosis. So she had received a complete clinical diagnosis, but the geneticist on the team, Dr. Austin Larson, had done a complete workup and found that there was a missing second mutation. And as I mentioned, this is a recessive gene. So 2 mutations are required to complete a diagnosis. So the second causative mutation was therefore missing. And it was that lack of a second mutation that had led to the Facebook post, which we had seen, and the plea from the family to find someone who might be able to perform whole genome sequencing to find the second mutation, to be sure of this really critical and devastating diagnosis. Now, the fact that the second mutation was not found represents a not uncommon finding in genetics. Approximately 10% of the time, we are unable to find the mutation that we know really ought to be there. And that's because 99% of the genome is so-called dark matter. It's intronic sequence outside of the region of standard clinical diagnostic labs interrogation. And so it's missed there. And so we decided to help the family look in those regions that had been missed by typical clinical diagnostic labs to see if we could complete this diagnosis for them. And there, what we found is that whole genome sequencing revealed a cluster of unusual reads deep inside an intron of the CLN7 gene. And that this was found in our patient, also inherited, we determined that it was inherited from her mother and absent from her father. And it was deep in intron 6 right here. And when Aubrey and I took a look carefully at these unusual reads, a closer inspection revealed to us that these represented chimeric reads. These were reads that in which a part of the read mapped to the human genome, and then the other part seemed to diverge into a sequence that was foreign and not recognized as part of the human genome reference. And in particular, there are two types of reads. In one direction, there was CLN7 sequence here shown in the gray, that then was fused to a foreign sequence, a poly-T sequence here, T in red. And the opposite sequence, there was native CLN7 sequence fused to a tandem hexameric repeat, what looked like a pattern of repeating GGG-AGAs over and over and over again. And that these two fusion events, these two families of chimeric reads, together represented a very interesting signature that indicated to us that what had happened in our patient was that she had suffered not a point mutation, a single letter change from one to another, but a DNA insertion that began with T's on one side and the hexameric repeat on the other side. The question is, where did this insertion come from? And there, scouring the literature and talking to colleagues, we, together with Alice Lee in our Division of Genetics and Genomics, we were able to deduce that this insertion represented an unusual mutation, a retrotransposon insertion, or a piece of jumping DNA, from a family of retrotransposons called the SVA retrotransposon family. And these are sequences that act in essence like genomic viruses. They hop in our genomes, they go from place to place, and they can cause disease if they happen to hop and land in the wrong spot. And when they do hop, they leave a footprint that represents their 5' and 3' ends because they insert themselves into their landing spots. And that footprint includes a poly-A sequence on one side and a hexameric repeat on the other side. And that these sequences here were the reverse complement of what we had seen in our patient. And so we were able to put all of these clues together to deduce that what had happened was that there had been the insertion of this SVA retrotransposon in her gene. Now, why am I telling you all of this? This is sort of esoteric information that is of intense interest to genome detectives like us, but doesn't necessarily point towards therapy, or does it? And here we asked ourselves the question, well, what is this insertion doing? This is deep in an intron, away from the coding sequence of the gene. So what impact actually does it have on the function of the gene? And here, again, reading the literature and hypothesizing what it could possibly be doing, we discovered and proved that this was a pathogenic mutation that actually alters gene splicing. We performed RNA isolation and did RNA sequencing analyses, as well as splice junction analyses and RT-PCR, which showed that the result of the insertion of this foreign retrotransposon in intron 6 was to introduce an abnormal splice site that led from exon 6, not into exon 7 where it's supposed to go, but into this abnormal site, which we named I6. And the use of this abnormal splice site led not to functional protein, but to an immediate stop codon that prevented the rest of the protein coding sequence, exon 7, 8, 9, 10, 11, 12, and 13, to be translated, and doomed this protein to non-functional destruction. And this, it turned out, was the in here. This insight that this early insertion was causing a stop codon, was causing a change in splicing, gave us an insight that offered us a chance to do something about it. You see, what was happening at the time was that we were reading about an antisense, a remarkable drug called Spinraza or nusinersen. And what Spinraza or nusinersen is an antisense oligonucleotide that had been just published in the New England Journal as a really promising investigational therapeutic for a condition called spinal muscular atrophy. This antisense oligonucleotide drug is a small stretch of synthetic RNA. It's artificially manufactured. And in this case, it's 18 nucleotides in length, and its sequence is shown right here. And we were reading about how this drug was given, when given by spinal tap to infants with spinal muscular atrophy, was capable of binding into the genomes of these individuals, and specifically to the mRNA sequence, the pre-mRNA sequence of a gene called SMN2, in such a fashion that it would alter the splicing of the SMN2 gene and allow normal SMN2 to be made, correcting an evolutionarily acquired mutation in SMN2. And that bit of molecular genetic trickery was having remarkable clinical effects, whereby children administered Spinraza or nusinersen were showing remarkably increased survival compared to patients who have spinal muscular atrophy, who unfortunately otherwise suffer neurodegeneration and pass away typically by age 2. And this drug was stopping that. And thinking hard about the success that we were reading about for this first in human class of drug, antisense oligonucleotides given to the central nervous system, we read more about the underlying technology, and we noted that when this kind of drug is given intrathecally, it's well absorbed by the spinal cord, but also by the brain. And it's present in not too steep of a gradient, so that you can get reasonable levels up into the brain that are only at the expense of having 3 times those levels in spinal cord. And that's a reasonably shallow distribution and shows that this drug can be delivered pretty broadly within the neuraxis. And then when you look at sections to ask in how many cells does it actually penetrate, one finds that there's broad neuronal uptake. If you take sections of spine or motor cortex or brainstem and look at neurons within those microscopic sections, you see that this drug, which is represented here in the brown dots representing antibody staining against this drug, this drug is found in nearly every single neuron in the field, indicating that this type of drug's delivery is very potent and very broad. With this in mind, we began thinking to ourselves, what if there were a way that we could take that technology with easy distribution via spinal cord injection and broad cellular distribution? What if there's a way that we could make Spinraza for our patient? What if we could make Spinraza for Mila? And going back through the literature and reviewing how that was done, we cobbled together a script based off of work that they had templated in 10 years of publications before us to develop a patient-specific screen whereby we could take our patients' fibroblasts, design a series of antisense oligos, and then screen them to try and restore normal gene splicing. So this work, which we accomplished in the ensuing year, rested on a number of different scientific pieces of evidence. First, we were able to use bioinformatics to design a series of splice-altering ASOs, looking at splice-modulating sequences near the mutation, and then target those splice-modulating sequences with antisense oligonucleotides. We were able to do our drug screening using cells cultured from our own patient, using fibroblasts, and then introducing exon-skipping antisense oligonucleotides into them by electroporation or by liposuction. And we could use our assays, simple assays at the bench, to look at measures of how much normal versus mutant splicing we were seeing. And we could calculate how much normal versus mutant splicing we were seeing, and then apply our drug and find out after application of a drug, could we actually boost levels of normal splicing? And we were able to find candidates that seemed to do so. And so our team, April, Aubrey, and Jay, using this approach, we're able to use this to advance a series of leads that were then characterized further using RNA-seq and transcriptome profiling. This is RNA-seq showing more complicated patterns of splicing between exons 5, 6, and 7, or the intruding mutation in our patient. And what one sees is that in the absence of treatment, our patient is only able to splice exon 6 to exon 7 properly 7% of the time. And a control ASO treatment did very little to change that. But our lead ASO was able to boost this by 3 to 5 times, up to 32% of normal splice events. Furthermore, we were able to advance bench work in collaboration with Joe Mazzulli to show that ASO treatment could rescue lysosomal function. That treatment of this with this oligonucleotide not only changed the RNA splicing patterns of our genes, but it changed the critical readouts that determined that the lysosome was healthier after treatment. And what one sees here are wild type cells under an electron microscope versus mutant cells from our patient, which showed that our patient cells had the abnormal accumulation of these large white bubbles representing storage vacuoles, abnormal storage material, protein and lipids built up inside of lysosomes. And the treatment of our patient cells with our lead oligo, which is called TY777 here, was capable of hugely reducing the number and the size of these storage vacuoles, really back to normal. And this work done in collaboration with Joe was also complemented by a number of assays that Joe had developed over the years to show not only electron microscopic improvements, but also a number of functional assays, lysosomal mass, autofluorescence, and autophagy, all corrected with treatment with our ASO. Finally, from a pharmacologic standpoint, we were able to demonstrate that our ASO drug in dose response curves showed potency similar to that established for the FDA-approved drug Spinraza. And these are curves showing that our drug was working in the low nanomolar range, which is exactly where you need to be for drugs with pharmacologic efficacy in vivo. All of this work was done in our laboratory under a compressed time frame given the clinical situation. We first met our patient in January 2017, and we completed whole genome sequencing a month after. We identified the splice disrupting retrotransposon in March. We began to discuss ASO treatment with the family in April. We generated patient cell lines in May and designed and began to order a series of antisense oligos in June. In July, we began the beginning of our first conversations with the FDA to talk about the path forward with this bench work that we were doing. More on that later. And in August, we were validating our functional assays. In September, we were showing and beginning to see evidence of restoration of splicing. And by October, we had evidence that lysosomal function was improving as well. And around this time in October, we were perhaps patting ourselves on the back, very pleased with the progress that we had made, but also really looking ahead, humbled by the amount of work that was remaining to actually deliver this to a patient. And here, just for context, one has outlined the normal steps that are involved in taking a bench concept that's established proof of concept and then bringing it into clinical trials and to patients. Usually there are many, many steps in between: proper drug manufacturing, release testing, formulation, animal tox studies, clinical protocol design. And this is an intense process that takes many, many years. Yet in our case, we were working under very constrained clinical circumstances. As fast as we had gone over those 10 months, the family was reporting to us that clinically our patient was declining rapidly, and she was losing milestones in many, many respects. And so turning to our team and to the FDA, this gave us an urgency and a motivation to move even faster. We decided to proceed to treat our patient, to seek permission to treat our patient under an individual patient expanded access protocol. And we were very lucky to recruit a number of ex-FDA advisors, Lauren Black, Mary Pendergast, and Sarah Goldkind, who with us helped us interact with the Division of Gastroenterology and Inborn Errors at the FDA, led by Program Manager Hong Vu. However, there were significant challenges in filing this IND, this investigational new drug application. In fact, this is the first time this particular route had been used for an entirely new drug. So there were a number of hurdles and precedents that we had to try to set. In tackling these challenges, we were fortunate to have the help of a number of folks in the oligonucleotide therapeutics community. I'd like to point out especially Art Krieg, who at the time was the president of the Oligonucleotides Therapeutic Society. And he arranged for us to go to the DIA FDA Oligonucleotide Therapeutics Conference, which was actually happening in October 2017, the same month that we were asking these questions. And there we had this amazing opportunity to interact with folks from industry, from the FDA, manufacturing consultants, toxicology consultants, and some of the best scientists in the field. And through that meeting and presenting to them our clinical dilemma, we were able to come up with strategies to tackle the many challenges that remain. And some of those challenges are listed here. On the manufacturing side, there was no applicable FDA guidance for cases like this. Like, how do you make a drug in months as opposed to years? And what are the safety requirements for testing at QC of such drugs? Our need for our patient was of such a small quantity that most of the standard GMP manufacturers weren't working on that scale. We only needed 10 grams of drug for decades of supply for our patient. But that was perhaps 50 times less than typical for many, many providers. We needed it a very rapid turnaround, weeks to months. And we needed it sterile for CSF injection. And the upshot is that the regulations for managing the manufacturing process, for the QC testing, for the formulation, and for the safety testing did not yet exist. In fact, today, I think they are still, this is an ongoing conversation. Just to give an example of how challenging all of this was, we designed a series of manufacturing campaign and had our drug manufactured. But then at the end of our manufacturing run, we hit a snafu. At the time we organized the campaign, we had decided that this drug could be released to us. We had negotiated this drug's release to us on a for research use only basis. And at the 11th hour, the lawyers involved on the manufacturer side came to us and said, that's not going to work anymore. We're going to have to label this not for clinical use. We said, wait just a second, that doesn't make any sense. We've been negotiating this strategy with you for months, and you know exactly that we intend to give it to a patient. That terminology can't be acceptable. What if we try something more reasonable? What if we call it investigational use only? And they thought about that, and they came back to us and they said, no, we're not comfortable with that. The best we can do for you is research grade. And we went back and forth like this several times, and we said, this is getting a little bit ridiculous. You know exactly what we intend to do with this. We plan to give this to a patient that we've identified, and how about we label it, investigational use limited to IND 136294. How about we label it for our patient? This is exactly our intent. Why are you throwing us a curveball at the last moment? And finally, we were able to agree on the following label, which was 2 lines and barely fit on the actual physical label that wrapped around the vial. But I say this not to throw those particular legal advisors under the bus, but just to illustrate, I think, how fundamentally uncomfortable everyone was working in this space, in this territory, where standards and questions like liability were not yet clear. Nonetheless, those hiccups aside, we were able to navigate those tricky waters. And we were actually able to complete a really aggressive manufacturing and animal toxicology campaign. When I say really aggressive, we were able to do this in just 3 months' time. And with a lot of help from Charles River Labs, from TriLink Biotechnologies, Brammer Bio, as well as a number of independent manufacturing consultants who volunteered their time to help us, Chantal Reed and Kim Tyndall, we were able to get through these necessary uncharted steps from October, November, December to release of drug to us in January of 2018. With the drug on the way, we were able to invite the family to come visit us and to move to Boston for an investigational trial in January of that year. We actually received the FDA permission to proceed 6 days after they arrived. And then 6 days after that, the actual drug shipments arrived and were put into the careful hands of Al Patterson, our chief pharmacy officer here at Boston Children's. And with that in hand, we were able to launch our trial in January of 2018. How did we do, and what is this case teaching us, and what has followed from this case? Well, I'd like to say up front that this was an extraordinary effort from many, many people, and the whole institution stepped up to help this young girl. And I wish I could say that this heroic effort from so many folks was a fairy tale story and led to complete resolution of this child's symptoms, reversal of her decline, and restoration to health. And unfortunately, that's not likely to be in the cards. We were working with a young girl with a really devastating condition who was already very far advanced in her condition. She had lost her vision. She had lost the ability to speak intelligible words, and she was largely unable to walk except with extreme assistance. And her decline over these last 3 years, we believe it's slowed, but it has continued. However, we were able to see significant effects of the drug on a few critical pre-specified domains that indicated to us that this effort was not for naught, and that significantly impacted the quality of life for our patient and for her family. And that's shown here in the seizure plot. This is a plot of seizures in our patient over the first year of treatment. Before treatment, she was having between 15 and 30 seizures per day. These seizures were not fun. They were lasting greater than a minute each, and they consisted of generalized tonic-clonic, uncontrollable movements of her extremities on both sides. And what we saw was that with dose escalation, as our drug was delivered, the frequency and the intensity of these seizures really took a remarkable decline. And I have to say, going into this compassionate use trial, this was more than I really had dared to hope for. But we saw the frequency of seizures go from 15 to 30 down to between 0 and 10 in those first 6 months. And we also saw the duration of the seizures drop from all lasting greater than a minute, 100% with these dark circles, to really none of them lasting greater than a minute. By this point, the family was reporting to us that they were lasting, most typical seizures were lasting a few seconds at tops. Now we began spacing out our doses, and we saw that when we moved from this careful but aggressive short dose intervals out to longer dose intervals, that the seizure frequency seemed to rebound a bit, but the intensity remained low. And when we adjusted our dosing schemes, and we did a reloading to bump up levels, increase the dose, and tighten up the interval. And we saw that we could eke out a little bit more benefit on the seizure side to reduce the frequency slightly further over the second year. And so it's data like this that indicate to us that while not a cure, and perhaps we were slowing progression but not stopping it and we're reversing it, that this data actually speaks, I think, to a pharmacodynamic effect of the drug on this important seizure outcome. And more importantly, the family tells us this represented a significant improvement in quality of life just from day-to-day management of those terrible seizures she had at the outset. All of this work was done in close collaboration with a really remarkable family that has been an incredible partner in all of this. There are a few families, I think, in a first of its kind trial like this would have been so ready to understand not just what we knew and what we didn't know, but just how uncertain we were about everything. And to be able to work through us and accept that uncertainty and do our best for this child. I think we were very lucky to work with this family and other families like hers. I think they set a high bar here. And this work really 3 years later, I like to reflect on what it meant for us to pursue this clinically and scientifically. And these were not easy decisions. This is not simple and clean bench work or bioinformatic work. But in working with patients, these are not clean experiments. But our decision to treat here, I have to say, was carefully thought through. And there were a number of factors that really compelled us to do so. First, we were working with a progressive and devastating disease. This is a condition which unfortunately exacts severe tolls in terms of medical interventions required and anti-epileptic seizure drugs. It requires intensive medical care for years, and it really takes a high toll on parents, siblings, and families. And what that meant for us was that the risks of what we were proposing, the treatment itself, had to be significantly weighed against the known natural history of this condition, and the fact that this is 100% of patients with this condition will pass away. Second, we were working with an identified single gene mutation with a very well scientifically characterized impact on genetics, on splicing, and a scientifically feasible fix. This is a single gene, a single mutation disorder. And this means that this genetically targeted treatment that we had devised arguably was addressing a root cause and arguably boosts the odds of positive impact. And that's an important point. I think in this day and age when we have exquisitely targetable therapies, these drugs are really precision guided and increase the priors to some degree of success. And that is important in thinking in how we think about them. Lastly, we were dealing with a patient that was, in this case, a true N of 1. Her particular mutation is not one that we've encountered in any other patient. And our drug is not applicable to other patients with Batten disease. It's specific to her particular mutation. What that meant was that our intervention had the flavor of medical care, not the flavor of a drug that we were going to market or commercialize. And this type of intervention, an intervention really tailored to a patient, meant that we were working with something that was much more akin to an individually consentable procedure. And that really truly mitigates the public health impacts of decisions that we make, that the FDA makes. They're not releasing a drug that's now going to be marketed appropriately or inappropriately to many other people. There is no market, there's just one. As I mentioned, this work, I think, for its unusual features, as I think I've outlined, it was seen fit for publication last year and attracted a lot of attention. I think that it received a lot of immediate attention because underlying it was this concept that we had exploited in this case that's really attractive to a lot of folks. It's of programmable medicines. What do I mean by that? Well, Spinraza or nusinersen is the 18 nucleotide sequence shown here. Our patient's drug is the 22 nucleotide sequence right here. It just differs in the number and the order of the bases. And what it has raised, it highlights a real world example of this concept that you could take a drug and customize it by changing the letters, keeping the same manufacturing process, keeping the same evaluation process, and keeping the same delivery, and customize it to many folks with many different diseases. And it's led to headlines like this: if DNA is like software, can we just fix the code? Well, let's think about that for a second. Could it possibly be as simple as that? Well, let's get to that in a moment. One clear repercussion of this work is that since the Mielsen effort, it spurred other N of 1 efforts. And this is clearly a trend which we're going to see more, we're seeing more and more cases like this in the field. I'm aware of half a dozen similar efforts right now, not counting our own in the space. Here's one from Colombia. This is case number 2. J-Suffusin is a drug for a young woman named J.C. Hermstadt who had an extremely aggressive form of ALS associated with mutations in the FUS gene. Her identical twin sister had died of it just 3-4 years prior. And she began showing symptoms approximately 2 years ago. In response to that, and I think partially also in response to early word and rumors that were getting out there about our work with Mealison, Neil Schneider and Bob Brown worked together with Ionis and Project ALS to take a drug asset which had been previously developed for FUS ALS and hugely accelerate its progress through the drug approval pipeline. And they were able to obtain compassionate use permission to begin administering this drug to JC using a preclinical program, manufacturing toxicology, which is designed specifically for her in mind. And that effort, which has actually now led to the treatment of several other individuals with FUS ALS in addition to JC, is a really important milestone that followed in the wake of our case. Of course, beyond these types of N of 1 efforts, there are many other traditional ASO programs in the clinic or in trials. And there's great promise in the ensuing years for using ASO approaches to target Huntington's disease, ALS, and, Alzheimer's, a number of other targets, Angelman syndrome. And this interest speaks to the promise of this space and this modality. However, more specifically to the idea of ultra rare conditions and even N as few as 1 types of situations, we at Children's have been receiving now, in the wake of this, hundreds of inquiries from rare disease families, clinicians, and foundations who may not have a large biotech or pharma working on their disease indication, asking whether or not this type of customizable approach might be applicable to their loved one or their disease. And I will say that from these, we've identified several dozen patients who, like Mila, have a very, very serious neurogenetic illness. And they have a mutation which looks like it may be ASO amenable. How do we think about these? And really what's being asked of us is, is Mealison a template for cases like this? Can ASOs be used as a platform for N of 1 or small n medicines? When we think about this, let me pause and talk about the word platform for a second. What are the characteristics of a drug that makes something a real platform? And this idea has been around for a long time. Monoclonal antibodies, any number of therapies are therapies for which we've had ambitions of turning them into platforms. But I think that ASOs are perhaps the only platform today in 2020 that really, or 2021, that really deserves that title, or it looks close to deserving that title. To be a platform, something has to be rapidly customizable. It has to be simple and inexpensive to manufacture. It has to be easy to deliver, and it has to be proven safe and effective. And arguably, for these various characteristics, ASOs are showing really great promise in delivering as a true platform. These first 3 items, I think, are sealed and delivered. They are rapidly customizable, very simple to manufacture. And at least for central nervous system indications, intrathecal delivery is quite uncomplicated. And safety looks very promising. Its efficacy and safety, of course, always require further data to buttress, but early indications are that safety is very, very good. And its efficacy is buttressed by successes like SMA, and I will say perhaps partial successes like Mealison, but many other very promising investigational trials, data which is rolling in as we speak. A word about the toxicology or about the toxicities. These are not consequentless drugs. These are drugs that do have known toxicities when they're given intrathecally. In fact, when you give them at very high doses in rodents or in monkeys, you can get symptoms in those rodents and monkeys that look a little bit like leg paralysis. Now that sounds quite concerning, and it should be. However, the saving graces of this toxicity profile are that 1, it happens at very, very high doses. 2, it's monitorable. To date, this type of toxicity has occurred along a spectrum, whereby at low doses you see no impacts, and when you get to very high doses, you can begin to lose reflexes in the hind limbs. And then when you get to, it's only at these very highest doses when you can get true leg weakness and incoordination. And then on top of that, this finding, when you do see it, so far appears to be reversible. That if you do venture into this territory at very high doses, when you have like weakness and incoordination, that waiting either hours or days, that effect does wash out, and there have not been permanent deficits observed in these paradigms. So that actually speaks to the manageability of and the predictability of this in clinical settings. Now, to use these drugs as a platform, what's our incentive, or what's our motivation here? What are we really trying to do? The problem that I alluded to at the very beginning of the talk is that there are 7,000 different genetic diseases. And the practical reality is that many of these are much too small for traditional commercial interest. The population sizes are just too small. We have ways to develop drugs if you can sell a drug to 10,000 patients. But if you have 1,000 patients or 100 patients or 10 patients, the structures that we've built as a society to develop those drugs can't sustain themselves. So what are we we need novel approaches to address those other conditions. There are massive unmet needs under the current system. So we need creative new approaches. What could you do with a platform for small N medicines like ASOs? Well, you can begin to meet these unmet needs, but you could also learn a lot of biology. You could use these efforts to validate gene targets and to probe critical therapeutic windows for diseases for which often there has been no attempt at therapy ever made. And an ASO approach, even for a subset of patients with an ultra-rare disease, would allow you to validate these targets, validate these timings, and pave the way and de-risk other approaches, including small molecule or traditional gene therapy. The trick in considering how to do this is to keep as few moving parts as possible, to keep constant, for instance, the chemistry of the ASO, the particular mechanism that you design the ASO to engage, like splice switching, the administration route, like intrathecal route, but just change the sequences. And as you do this and explore a diversity of ASOs that all share similar common structures, we need to share data broadly so we can aggregate the experience with this class of molecule and build data to support this platform and support this approach. We have, over the last 2 years, been incubating and executing on essentially a pilot proposal to do exactly this, with the goals of helping patients in a responsible way, educating them about the risks, to gather this data on safety, tolerability, and efficacy, and to do these things that I've described, probing these windows, piloting outcome measures, and so forth. And along the way, we have to be pretty careful too, because we also know that with possibilities like this and tools like this, we can't have John Wayne's, right? We have to be very careful not to raise expectations too quickly, too broadly, too confidently, because what drives us to work with this patient population is that they have huge unmet needs, but that makes them vulnerable. And so trying to find the best ways to present this and inform patients about exactly what this new aggressive approach looks like and what risks and potential benefits are entailed, I think is something that will occupy us for a long time. Here's an example of another program that we have proceeded with in the line of the Nielsen program. We met a 2-year-old girl a year and a half ago with a condition called Ataxia Challengertasia. This is a young girl who was born on newborn screening and noted to have a low TREC count, which is a part of the statewide newborn screen that flags children who might have a severe combined immunodeficiency. She was worked up by an expert group of folks at Stanford, including Jennifer Puck, Audrey Foster Barber, and John Day, who astutely pointed out that this is not severe combined immunodeficiency, but actually what she had was recessive mutations in a gene called ATM. ATM is a cause of a neurodegenerative disorder called Ataxia Challengertasia. It does have a slight immunodeficiency component, which is why it showed up on this SCID screening, but that's actually of little clinical consequence. More importantly, however, AT is a condition associated with cerebellar neurodegeneration. And while as a newborn she was fine, patients with this condition begin to experience loss of coordination beginning around age 3, 4, 5, and progressing to the point where by age 13, they're in a wheelchair. And by their 20s, they may pass away from complications of having a lack of cerebellar function. It's a progressive and sadly fatal condition. And there are no treatments available for it. This patient who had been identified as at birth was lucky because she, it turned out, had a mutation which we analyzed and recognized to be a splice-altering mutation, not unlike Mila's. And after she was referred to us by the AT Children's Project and her clinicians at UCSF and Stanford, we generated cellular models of her disease, designed and evaluated a series of drug candidates to target her splice mutation. And we were able to identify ASO hits and validate that they worked, optimize them, and then obtain permission from the FDA to manufacture, safety test, and then begin treating our patient with this otherwise untreatable condition in just 18 months' time. And she is coming on her first year of dosing right now in January of 2021, because we started about a year ago. This work was done, as I mentioned, in collaboration with the AT Children's Project. And it's symbolic of the type of opportunity that the Mealison Project has opened our eyes to. And one question is, how many other patients are there out there who might benefit from these investigational attempts or might be eligible for these investigational attempts? And in an extension of our collaboration with the AT Children's Project, we've looked through a whole genome sequencing data of a whole large cohort of AT patients. And we find that actually up to 1 in 5 of these affected individuals may have a mutation that looks like it's amenable to treatment with a splice-altering ASO. This diagram here just shows that we reviewed 235 cases in this cohort and found that 9 to 12% of these had mutations that are at least medium confidence ASO amenable. And up to 23% of them have mutations that look possible for ASO intervention, although more work will have to be done to firm that number up. Along the lines of our work with AT, we have also put forth and opened a clinical investigational trial for a 4th case in September of last year, a case of epilepsy of infancy with migrating seizures, in a collaboration with our lab and Purdue's lab here at Children's and a Rare Disease Foundation. And there are many more cases like this along the way that we are studying carefully to see whether or not there are opportunities for intervention. Now, I'm going to close here with just a few final thoughts because I'm well at my time. And it goes without saying that there are really important ethical and regulatory considerations that need to guide this work. When we published our article describing Mealison in our Batten patient, it was accompanied by an editorial from Janet Woodcock and Peter Marks, the head of the Center for Drugs and the Center for Biologics at the FDA, describing the impact of this case on regulatory precedents and the need to develop new regulations, new guidelines for this unusual scenario, which is going to become only more and more common. And as we think about these regulatory implications, we also have to think about ethical considerations and practical considerations like costs. And these are research interventions at this point. So it's too soon to put a price tag on it. These are not services that we're providing, but research. But it goes without saying that this type of research is very expensive. And while there's much room for the costs of this type of development to shrink, at the moment, when the costs are high, being very careful about patient selection and thinking about how we distribute the opportunities to enroll in these sorts of trials in a fair and just way is really, really paramount. To help navigate these, we are committed to working with the FDA to establish a safe path. The FDA, as you know, releases guidances to describe to the community its approaches towards tackling tough issues like rare diseases in general, severely debilitating or life-threatening diseases, and so forth and so on. but there's no specific substantive guidance yet for how our particular situation. We're very pleased, though, that our conversations with the FDA are leading to, we've gotten real firm commitments from the FDA that they are working on this, and the first fruits of that came out just a couple of days ago. January 4th, their FDA's first business working day in 2021, they announced the first procedural guidance relating to IND submissions for individualized antisense oligonucleotide drugs. And while this guidance focuses just on administrative and procedural recommendations, and it's the beginning of a conversation, there's much more meat to be added to this about specific standards for proof of concept studies, toxicology, manufacturing. But it's the beginning of a very important conversation. Comments due in March. If you're interested in this, please investigate and join that conversation, add your voice to it. In addition to the FDA, we're working with other stakeholders, the Oligonucleotides Therapeutic Society. Here's Art Krieg. Here's the Annemieke Artsma-Roos, who followed in Art in his footsteps as the president of the OTS. And we've also engaged with important industry players like Frank Bennett and Stan Crook of Ionis. Ionis has launched their own charitable foundation to support work like this, work like Mealison. And working together with these folks, we can come up with inclusion criteria and clear goals and delineations of what we are trying to accomplish in these early investigational days. All of this is motivated by an opportunity to really fire test what could be, what we hope could be a true therapeutic platform for accelerating drug development and extend the reach of our system to many patients with rare disease that currently aren't being served. And antisense oligonucleotides are that platform right now that deserves this attention. You know, down the road, could AAV gene therapies come to that point? Could CAR T therapies be an example of that? Cancer vaccines, CRISPR genome editing, many other modalities will aspire to that platform role. But I think the important work that we do to establish some of the ground rules for at least one modality will extend to these others as well. My very last slide is to just speculate a bit as to where this is all going and what this type of landscape looks like. And I will say at the beginning, as I mentioned for Mealison, that our work on Mealison was not exactly like a traditional marketable drug. And many would argue that it wasn't purely a procedure either. It's something of a hybrid between the two, but who knows how this will all shake out. One thing that's clear is that in these early days, this work is investigational, and one possibility is that it will never leave the investigational stage. And in fact, there are some drugs, some therapies that we offer to patients that remain in this purely investigational phase. They're never marketed. There are drugs for snake antivenins, for instance. The market for that has never been large enough to sustain marketing approval. But these INDs have been opened for years and years and years, and they're used to treat patients successfully. And that could be a future way that this field develops. It's interesting to think, however, might the FDA approve something more than that? Might they approve a process for making customized drugs? Could you have multiple drugs under a single manufacturing and testing paradigm? And that you could roll out new versions of it for different patients at different times? And arguably CAR T and cancer vaccines are an example where they already have done something very much like this. A third model to consider, though, is that perhaps this is not a traditional drug. Perhaps as a surgical procedural model, where this type of treatment could be offered as medically innovative care. And here, I think a prior example in the literature is something like bone marrow transplants or other organ transplants. These are intensive, resource-intensive interventions that have strong medical value, but aren't delivered in the same type of way that we think of in a traditional FDA-approved fashion. And there are some parallels there too that are intriguing to think about. So I'm going to conclude here and thank you for your attention. Thank I'd like to extend a heartfelt thank you to our first family with whom we partnered in all of this work. Our work with Mila, for Mila, I think we hope will extend to offer hope and investigational options to many other children beyond her. And I've been mentioning the support of many collaborators and funders along the way. This is the slide showing the huge number of folks at Children's and elsewhere that helped with the Nielsen program. And this slide shows the many folks that were involved in our AT program and other projects that I've mentioned along the way. Thank you for your attention, and I'll happily entertain any questions and feedback you have.
That's fantastic, Tim. Tim, if you stop sharing for one moment, I will share. Let's see. And reminding people to put their questions in the chat box. So, Tim, this is absolutely spectacular. Let me pick up with something that you were really getting to at the end, but which is a fascinating idea of sort of innovative medical care versus an FDA-approved therapy. And you're absolutely right, of course, that surgical procedures may be extremely innovative and never undergo those kinds of reviews. This feels more like a drug. So how are you conceptualizing where the boundaries would be if we're actually creating an injectable, intrathecally injectable substance here that looks a whole lot like a drug in a drug manufacturing process, but is not a drug? That's really fascinating.
That's a great question, Ken. And I think the way to kind of discuss that really interesting question is to think about what are the things that make this feel like a drug. It is a drug. It is a drug. But what are the actual, let's dissect that a little bit and think about what is it that makes it different than a novel coiling procedure for a cerebral aneurysm or something like that. And I think the features are this. I think the features are that it relies on the successful design and delivery of these drugs does rely on expertise that currently doesn't live within medical, surgical, neurological, genetics divisions. That we had to reach out to manufacturers and toxicologists who live in industry to guide us and to give us hints, tips, and to help with us with the design of our protocols to successfully vet our drug prior to delivery to a patient. And so one is just simply the practical fact that there's expertise that doesn't live on a medical team currently, right? The way that a neurosurgical team can pull together everything it needs to do what it needs to do. The second thing is that it involves know-how about the chemistry and actual experience that is privileged information, because there are many other companies working on ASO drugs. They hold information about their biodistribution, their PK, their safety profiles, how they behave when they're manufactured in certain ways. And that's proprietary information that is those companies, it's owned by those companies. And on top of that, the FDA also has databases of IND submissions for other ASOs that they've received. And that data is all relevant to these efforts. The FDA sits in a privileged space of having access to this information that it would be like the FDA having this side pot of surgical procedural knowledge that only they had access to, that they felt compelled to be able to deliver to surgeons to help improve practices, right? So I think that creates some, those are the characteristics that kind of make it different. And I think that the one approach that we've been thinking about to address that is that, yes, it is a drug, but yes, its flexibility, and its customizability and the opportunity here really impels us to find ways of sharing that information, to create collaborations and consortia by which that type of information, which is normally kept proprietary, can be shared, such that it can be used more like a procedure. And in the service of patients who, frankly, are just 100 patients are never going to be served by the current funding models. There's no way to recoup an investment required to treat a disease where there are only 100 patients. Well, maybe we can declare that a pre-competitive disease, like a pre-competitive space, where industry, FDA, investigators can form a consortium and share the know-how necessary to treat those patients.
Would that presume in some way that one of these things could never become a drug? In other words,
yeah, right.
if you're going to be pre-competitive,
Right.
is someone going to fake to the left?
Right. I think that's the art. The art is setting it up in such a way that you can create the incentives without creating loopholes that allow people to game the system, right? And I think that if you're really talking about a disease that there might be only a few dozen patients, you're probably safe. That there probably aren't ways to say you're going to do this and then try and blow it up and commercialize it. You know, it's when you start talking about drugs that maybe are in the few hundreds range, then it gets trickier, and you worry a little bit more about that sort of possibility. But I think that if focusing on the rarest conditions, one ought to be able to proceed safely in that space without too much worry of that type of misbehavior.
Great. So Tim, there's a bunch of questions in the chat. Let me let's rattle through a few of them. So the first one that went in, and this was before you talked about the hind leg weakness of the mice and the toxicity trials, was what are the known anticipated drug side effects? So, and safety concerns. Since you addressed that a little, let me just, but that it is an interesting area, let me ask, what does high throughput toxicology look like in this pipeline, and how do you know what to look for?
Great. So let's go through the known side effect profiles. So if you look at the label of a drug like Spinraza or nusinersen, one that's a label that's been negotiated by Ionis with the FDA, you will find on it things to do with blood clotting and thrombocytopenia. You will find on it proteinuria, so kidney problems and platelet problems as being two prominent potential side effects, right? And the reality is that those are true known class effects of these drugs when they're given IV. When you give them intrathecally for central nervous system purposes, the drug is so efficiently taken up by the brain that you can get away with tiny, tiny doses such that, practically speaking, those sorts of labeled side effects should not be a concern. That's our current understanding, that those side effects should not be a concern. So what you're left with are these potential neurologic complications. And again, these only occur at high doses. Really, the trick here is that this is, I'm not going to say complete, it's certainly not completely unexplored territory. There are some companies that are making really big investments in CNS active ASOs. And the question is, how good can we get at predicting toxicology? How good can we get at predicting the effects here? And high throughput screening to teach us about how to predict these effects. You can imagine the experiment. You can imagine taking a combinatorial series of ASOs with slightly different sequences and injecting each of them into a dozen mice, a dozen rats, a dozen monkeys, and using those systems or a selection of those systems as a way to screen for which ones are bad and which ones are tolerated. And since the sequence of these is easily computationally described, you can use that data that you get to build up models for what sequence motifs confer which properties. And that is a really enticing experiment that is happening, I know, in industry right now, and learnings from that need to be derived. And how tractable that computational problem is going to go a long ways towards determining the velocity of the space.
Great. Here's another question. How do you perform the identification of ASO hits? Are there computer programs that you use to optimize the ASO for a specific splice mutation?
Right. So when we started this, we had no prior experience doing this. So we took a fairly naive approach. And it turns out that that naive approach, I shouldn't say it's naive. I should tip my hat appropriately to people like Adrian Craner, who had actually designed sort of biologically principled based approaches towards where to put ASOs to have certain effects. When we began looking for splice modulating sequences in melicin, we were able to use a number of splice predictor sites, looking at max end scan scores and human splice finder and a number of different algorithms to predict which sites were likely to be either inhibitors or enhancers of splicing. And then we were able to design ASOs against those regions and found that by really restricting our search space to those regions, we were able to find pharmacologically active ASOs. So that actually, that rather simple approach works really beautifully. You know, you can also empirically tile an entire region and do it, you know, experimentally. And chance, you know, that gives you the chance to find sequences that are not as well predicted as by the algorithms as others. But I think that's a tractable problem that there are tools to address.
Great. Here's one. We are working on engineered food grade bacteria to treat very early onset IBD in children. How do you see programmable bacteria in this field?
Oh, that's very cool. I'd love to learn more about that.
That's Zoltan.
I think that the parallels here are really in the regulatory approach. I think I can't really speak responsibly about scientific connections and about programmable bacteria here, but I think really what's critical is the idea that the FDA, and they recognize this, Janet Woodcock recognizes this, they need a way to think about therapies that work really well, but have to be customized to each individual case. And right now, the existing guidelines and templates for submitting investigational new drug applications are derived from the idea that you're going to submit a small molecule, maybe you purified it from a natural substance, and then you're going to give that same drug to 30,000 people. And there's one IND filing, there's one molecular structure, the chemists look at that one molecule and they study that in safety tests. And now you say, well, no, I've got a process for making ASOs, programmable bacteria, whatever it may be. And I have a process for evaluating safety, and I have a process for customizing it to each patient. They recognize the need to develop the flexibility to handle those cases. And I think that that's the shared commonality here. I think that's, you know, where our work is going to intersect with work like that.
Great. Here's one wonderful talk. Can you comment on the potentials and challenges of developing precision mRNA therapeutics in order to restore the expression of target genes, which seems to be a good complement to the ASO therapeutics that reduce target gene transcript expression?
I think that's a wonderful technology. Hats off to Moderna and many other companies that are pushing that field forward. I think there are a couple of things. So the beautiful thing about mRNA therapy is the incredible broad applicability it has. As a gene replacement strategy, there's nothing more satisfying than saying that, you know, here's a patient with, take patients with Batten disease who have no functional CLN7 gene function. You can treat 100% of patients if you provide CLN7 mRNA to those patients. The places where that technology will have to be optimized to really fulfill its potential are delivery and dose. Delivery, how do you get it into the right cells? And how do you get it into, and how do you keep it out of cells that you don't want it in, right? When you deliver an mRNA, you may be bypassing regulatory mechanisms that could be important for restricting where it's expressed, at what levels it's expressed, and so forth and so on. And so you're really relying on getting the dosing right so that the cell gets enough. And so those are those two problems, delivery and dosing, are the challenges to be solved. And you could say the same for AAV-based gene therapy and so forth for mRNAs. But otherwise, it's an incredibly, there are probably cells and tissues in which those types of problems are relatively low on the scale, and where you could begin using mRNA, synthetic mRNAs as a platform therapy right away. And that's really exciting.
Great. And one last question. How do you see the long-term monitoring of toxicity, particularly across groups of these oligonucleotides that may have similar effects?
Yeah, absolutely. So the opportunity and the appeal of doing this type of work is the ability to exquisitely tailor a drug to a particular patient's individual mutation. And the challenge is just like trying to be able to aggregate sufficient data so you get signal to monitor those really critically important things. I think that to the extent that this work will, it's very clear that there's intense pressures to investigate this idea further. And if those investigations produce data that is permissible, this is the type of work that's going to have to be really carefully coordinated across many clinical sites, because looking for safety signals across a class of drugs in different patients with different diseases is an insanely complicated problem. And one that we need to build up our chops in terms of establishing what's necessary to build learning healthcare systems. We need to be able to capture how these patients do and pool that data efficiently to be able to track the success or failure of this approach. And that's not an insignificant challenge, but it's a really important one. And it's not specific to ASOs. It's true for any individually tailored therapy that we can imagine is coming down the pipeline. For us to be able to track individual specific drugs and look for individual specific toxicity, we're going to have that capability.
Fantastic. Tim, this has been truly inspiring. One of the great translational successes of the last many years. Congratulations. And taking us through the mechanics, the thought processes, the collaborations that are necessary to begin to deliver true precision care is inspiring. It's also sobering, as the term gets thrown around a lot. And so you are here where the rubber meets the road. Thank you for what you're doing and for sharing with us this past hour. It's been really great, and I hope you'll come back and update us before too long. So I just want to post to the group listening. We've got upcoming talks. Atul Butte, who's actually a graduate of the Boston Children's Hospital Computational Informatics program, but who's now the Chief Data Science Officer for the whole UC system, who is doing incredible work at essentially California scale, which is a big scale, in precision, achieving precision through information sharing. Lawrence Lessig, who founded the both Creative Commons, an alternative to copyright, and also the Electronic Frontier Foundation, also writes a lot about democracy, and interestingly was, in fact, a presidential candidate himself in the 2016 election. Enrico Coiera, one of the really founding figures in biomedical informatics, particularly at the intersection with sociology. Michael Creamer, who won a Nobel Prize in economics for his work on randomized approaches to studying interventions in under-resourced global settings. And then Chrissy Farr, health tech investor at Omers and Ventures. Some of you may have seen her ubiquitous byline in the health and health IT and IT segments on CNBC, where she just switched over into her new health tech investor role. She's spectacularly charismatic and informed, and I'm sure that'll be a great talk. We've got more coming into the scheduling as well. And there was one more question in the chat, which is, do we post recordings? And yes, we do. On the on the chip.org website, you can find links to those recordings, and Tim's will go up as well if you missed any part of it. So giving folks 4 minutes back, and thank you very much. We'll see you in February for Tool Buttes talk. Thank you.